Good morning everyone. In the previous section I have discussed the nervous system, what is the nervous system, and then the neurons. The nervous system is made up of the neurons, the specialized cells. Nervous system is made up of the specialized cells and that is called the neurons. And the nervous tissue consists of also the supporting cells. The supporting cells and that is called the neuroglia cells. The neuroglia cells, the supporting cells and that is called the neuroglia cells. And neuroglia cells plays a supporting role in nerve tissue stroma, and the neuroglia cells, these cells and their process fill up the space between the neurons. The neuroglia cells, it provide insulation to the neurons and their process. It provide insulation to the neuron and their process. What is the size of the neuroglia cells? The neuroglia cells is much smaller than that of the neuron. However, in the central nervous system, the number is 5 to 50 times more than that of the neurons. Now, the neuroglia cells are capable of multiplying in a mature nervous tissue. They are capable of multiplying in a mature nervous tissue. If there is the injury, or if there is the disease of the nervous tissue, the neuroglia cells multiply to fill the space occupied by the neurons. If there is the injury, neuroglia cells multiply to fill the space which is occupied by the neurons. What is the difference between the neuroglia cells and the nerve cells or neurons? The first is the size, second is the number, and third is the impulses. The neuroglia cells, they are much smaller than the neurons and the number are 5 to 50 times more than neurons. The impulses are unable to generate nerve impulse. The neuroglia cells unable to generate the nerve impulse. They are unable to generate the nerve impulse. These are the two differences between the neuroglia cells and the nerve cells. Now, the neuroglia cells, they are divided into the two types: the neuroglia cells of the central nervous system and the neuroglia cells of the peripheral nervous system. Then what are the neuroglia cells of the central nervous system? First is the epidermal cells, second is the astrocyte, third is the oligodendrocytes, and fourth is the microglia cells. Neuroglia cells in the peripheral nervous system, first is the Swan cells and the satellite cells. If we see the structure of the neurons, the neuron have the myelination in the area of the axons, and the oligodendrocytes and the Swan cells, it forms the myelin sheath. They forms the myelin sheath. Oligodendrocytes, it forms the myelin sheath in the central nervous system, and the Swan cells forms the myelin sheath in the peripheral nervous system. Now I will discuss the different cells of the neuroglia cells of the central nervous system. First is the epidermal cells. What are epidermal cells? Cells are arranged in a single layer. And they are cuboidal or columnar in shape, and microvilli or cilia are present on their apical surface. The epidermal cells, it lines the ventricle of the brain. And the central canal of the spinal cord. I have shown in the figure the epidermal cells, the cilia and the cytoplasmic process. What is the function of the epidermal cells? They are responsible for formation of the CSF and assist in its circulation. They are responsible for formation of the CSF and assist in the circulation. Now, next is the astrocyte cells. What is the astrocytes? They are so called because they are star-shaped. The astrocyte cells are star-shaped. Embryologically, astrocytes, it is developed from the neural crest cells, and they are of two types. One is the protoplasmic astrocyte and second is the fibrous astrocyte. The astroplasmic astrocytes have thick process with abundant granular cytoplasm, and fibrous astrocytes have long and thin and straight process, and cells are present only in the white matter. What are the function? Astrocyte cells, function of the astrocyte, it helps in providing the nutrients. To the neurons and helps in formation of the blood-brain barrier. Figure showing the protoplasmic astrocytes. Next is the when the neurons or nerve tissue are damaged, the astrocyte cells are undergo hypertrophy and hyperplasia to form scar tissue, hardened scar tissue. So what is hyperplasia? Increase in the number of the cells, that is the Plasia. And what is hypertrophy? The cell size increases. If the cell size increases, that is the hypertrophy and hyperplasia, which also occurs in the case of the uterus. In the case of the uterus. Now the hypertrophy and hyperplasia, that leads to the formation of the hardened scar tissue, and this process is called astrocytosis, or also called the glasses or the sclerosis. This figure showing the fibrous astrocytes. They have long and thin and straight process, and cells are present. The cells are present only in the white matter. They have long and thin process, and cells are present only in the white matter. The protoplasmic astrocytes are present in the gray matter, and the fibrous astrocytes, they are present in the white matter. They have long, thin and straight process, and cells are present only in the white matter. These are the difference between the protoplasmic astrocytes and the fibrous astrocytes. Next is the oligodendrocytes. What is oligodendrocytes? They are the small round or oval cells. With only a few cytoplasmic processes. Figure showing the oligodendrocyte, and they have a few protoplasmic processes. These are the processes, and there are the few protoplasmic processes of the oligodendrocytes. And oligodendrocytes develop from neural tube. They are developed from the neural tube. What is the function of the oligodendrocytes? It produces a myelin sheath around the axon in the central nervous system, not in the peripheral nervous system. It produces the axon. I am showing the axon. This is the axon, and it produces the myelin sheath in case of the central nervous system. Next is the microglia cells. What is microglia cells? They are involved in phagocytic activity. And they are mesodermal in origin. And its function is the removes the microorganism and dead nervous tissue and foreign matter by the phagocytosis. Now the Swan cells, they are flattened cells. Next is the Swan cells. Microglia cells removes the microorganism and dead nervous tissue from and foreign matter by the phagocytosis. Next is the Swan cells. What Swan cells? They are the flattened cells with flat nucleus, and it's surrounded by the cytoplasm. And these cells are present in the peripheral nervous system. The Swan cells are present in the peripheral nervous system only. Now the astrocytes, the epidermal cells, the microglia cells, and oligodendrocytes, they are present in the central nervous system, and the Swan cell and satellite cell are present in the peripheral nervous system. This is all about the neuroglia cells, the epidermal cells, the astrocytes, the oligodendrocytes, and the microglia cells and neuroglia cells of the peripheral nervous system, that is the Swan cells and satellite cells. Thank you.